Welcome to this tutorial for MolD 1.4. MolD, an acronym for molecular diagnosis, is a bioinformatic tool designed to identify DNA-based diagnoses for taxa. DNA-based diagnostic characters can enhance the formal description of taxa, and reporting them is important to satisfy the requirements of nomenclatural codes in cases where no reliably diagnostic morphological character states exist to define a new species. First, let's quickly review how DNA characters can vary within an alignment. This slide shows part of a DNA alignment of the barcode fragment of the cytochrome oxidase subunit 1 gene for a set of marine mollusks of the family Conidae. Invariant sites are shown as dots, and the four nucleotides A, C, G, and T are shown in the usual colors green, blue, black, and red. The top rectangle shows the sequences of the query, in this case the genus Conosprella, and the bottom box indicates the reference taxa. Our goal is to find a set of diagnostic DNA characters for Conosprella, the query taxon. Let's see how we can differentiate the query taxon from the reference. First, we note that there is a specific site at which all members of the query are different from all reference group members, site 256. Therefore, in the given alignment, this single site is sufficient for diagnosing the query taxon Conosprella. We refer to this as a type 1 site or type 1 character. There is only one site like this in the alignment. However, there are many sites where the DNA letter is conserved across the query sequences and differs from at least some reference sequences. These sites are shaded in gray here, and they are the ones used by MolD alongside the type 1 sites to propose DNA diagnoses. MolD takes advantage of the fact that such sites, which we call type 5 sites, can be combined to become diagnostic. We term this a DNC, which means a diagnostic nucleotide combination. For example, the combination of T at site 266 and A at site 286 is unique for Conosprella. The same is true for the combination of nucleotides that we see at sites 286 and 295. Therefore, each of these constitutes a minimal DNC, or MDNC, meaning a proper though minimal molecular diagnosis of our query. Minimal here refers to the fact that a possible mutation of this site in any query sequence immediately disqualifies the combination, making it not any longer diagnostic. So the question is, can we make a DNA diagnosis more robust so that it accommodates nucleotide replacements in query or reference taxa, but still remains unique for the query taxon? In MolD, we achieve this by constructing a redundant or robust DNC, abbreviated as RDNC. This is a combination of nucleotide sites that is not minimal but redundant. In other words, it comprises a larger number of informative nucleotides, aiming for increased robustness. The upper line of this slide shows an example of an RDNC, constructed from the previously identified type 1 and type 5 sites in our alignment. This was a brief introduction to give you an idea how MolD assesses DNA characters and the type of output it generates for DNA diagnosis. The actual algorithms for calculating MDNCs and RDNCs are not trivial and are explained in the original publication introducing MolD. Now, let's see how MolD works in practice. Here, we illustrate the use of MolD through the standalone executable distributed in the iTaxoTools project. Web-based versions of MolD can also be found on the iTaxoTools website. Advanced users may prefer to run MolD in command line mode directly from the Python source code. The second panel allows you to choose the preferred method for setting parameters. You can open a FASTA file containing the alignment of sequences you want to analyze by clicking on Open, or by selecting the input file using the Browse button in the Sequence Data File panel. The interface also includes two panels dedicated to selecting taxa for one versus all and pairwise diagnoses, enhancing specificity in analysis. Finally, the program provides 4 blocks, allowing fine-tuning of parameters. Notably, these advanced settings come with reasonable default values. The first thing to do is to load a properly formatted alignment file. The simplest option is a FASTA formatted file, where each sequence header comprises two strings separated by a pipe character. An example of this is illustrated in the box for one of the sequences. The first part of the sequence header, highlighted in green, represents the specimen ID, while the second part, in blue, serves as the taxon identifier. If you have any doubts about the format, please refer to the example files distributed with the program. It's crucial to note that the queries diagnosed by MolD correspond to the taxon identifiers. Therefore, ensure that all sequence headers are correctly formatted, containing only one pipe symbol, and that taxon IDs are consistently spelled across sequences. To maintain clarity, avoid using special characters in the headers, opting for standard alphanumerical characters and underscores whenever possible. For converting sequences and metadata curated in a table format into this specific FASTA format, you can utilize the DNA Convert tool, also included in iTaxoTools. After loading your alignment file, the remaining settings can be left at their default values. This way, MolD will attempt to identify diagnoses for all taxa in the alignment. Press the Run button, and the analysis starts, showing the progress in a newly opened viewer field. This usually takes some time, depending on the number of sequences and taxa in your dataset. Once the run is completed, MolD shows a notification: Molecular diagnosis completed successfully. By pressing OK, you access results of the run. You can either save all the output files by using the upper Save All button, or you can use the Save or View buttons to inspect or save only the files of interest. If you want to repeat the analysis with different settings or start a new analysis, simply click the Clear button to return to the starting window. In case you encounter issues at this stage, such as memory constraints when analyzing a very large file, you may need to close and restart the program. Moving on to MolD's advanced features, you can adjust many parameters directly through the interface. Alternatively, you have the option to upload a configuration file for precise settings replication in future analyses. Refer to the MolD manual for details. In certain scenarios, you may want to diagnose specific taxa rather than all of them. In such cases, the From List option enables you to select your target taxa. You can enter the taxon IDs to be diagnosed as a comma-separated list or request diagnoses for all taxa with more than a certain number of sequences. Additionally, you can combine taxa, such as closely related ones, to diagnose more inclusive units. For further details, refer to the manual. Another option in MolD is to treat gaps in the alignment, originating from insertions or deletions in some sequences, as a separate character state for the purpose of diagnosis. Alternatively, they will be treated as an ambiguously called nucleotides, equivalent to an N. Be careful with setting gaps as a separate character state if some sequences are incomplete at the beginning or end, and this missing data is coded with dashes, similar to true gaps. Ideally, use alignments where all sequences are complete. In the two lowermost sections of options, you can modify and adjust the parameters for MDNC and RDNC recovery. Regarding MDNC-related parameters, you can adjust the following. First, the number of informative alignment sites considered by MolD corresponding to the option Cutoff. Second, the number of ambiguously called nucleotides allowed in a sequence corresponding to the option Number N. Third, the number of MolD iterations, where each iteration results in one DNC. Fourth, the starting and final lengths of the MDNC, corresponding to the option Maxlin1 and Maxlin2. And finally, designate one sequence as the indexing reference, and the indexing of sites will be adjusted to the start of the reference sequence. In the last panel, you can adjust parameters for RDNC testing against simulated DNA alignments. For details, refer to the MolD manual. The robustness of the resulting RDNC increases with a higher number of modified sequences and stricter scoring. However, it also raises the chance that no sufficiently robust RDNC can be recovered. At the end of this tutorial, let's take a quick look at the output files. You'll find a log file containing all standard output and two diagnoses outputs. The first one includes the diagnoses of the selected taxa versus all others in the dataset. As shown on this slide, it starts with the parameters section, followed by taxa blocks in alphabetical order. Each taxon block begins with the number of sequences analyzed and concludes with a phrase corresponding to the identified RDNC for that taxon. This sentence can be directly copy-pasted into a taxonomic paper, if desired. The second diagnosis output file comprises simple two-way pairwise comparisons between taxa, which can be relevant for descriptive purposes. It is particularly useful in exceptional cases where no RDNC can be recovered. This file includes lists of all sites where one taxon differs from another, citing the specific DNA character states for each of these sites. For further interpretation of the results, refer to the original publications of MolD and consult the documentation and help files on the iTaxoTools website.